Hello, everybody. We're going to finish chapter one. Essentially, what we're going to do is we're just going to clean up a little bit in the first section of chapter one. We'll talk about evolution and taxonomy, which is in 1.2. Then we'll just touch really quickly on the scientific method. I know that you did a lot of that in middle school and previously in science classes. So we'll just watch a movie essentially for that. Again, my goal is not to bore you or to rehash the entire chapter here. It's just to give you a little bit of insight on things that I think are interesting or that kids may have misconceptions on. Let's start with the movie.
Lactose tolerance evolved about 10,000 years ago. Over the last century and a half, the average height of a human being has increased by roughly 10 centimeters, and within the last 6.5 decades, the global lifespan has shot up nearly 20 years, largely thanks to science. Breakthroughs in science have propelled our species forward. In fact, National Geographic and TV's new series, Breakthrough, covers some cutting-edge innovations that are happening today, from biology and technology merging to solutions for our water crisis. But what will humanity look like in 1,000 years? It's already hard enough to remember a world without smartphones, but scientists predict that within the next few decades, computers will reach the computational speed of the human brain. Not only will they be able to speak and interact, but listen and remember. This also leads to a philosophy known as transhumanism. In the future, nanobots, or tiny robots, will be suddenly integrated into our bodies, enhancing our own abilities. No longer will we be limited by our own physiology, but truly become a mixture of biology and machine on the inside. Ever heard of utility fog? Imagine a cloud of countless microscopic robots which can be rearranged into virtually any configuration nearly instantaneously. Buildings could be constructed as necessary and disassembled when space was needed for something else. Picture your house disassembling when you leave in the morning so that space can be used for something else. With increased urbanization and globalization, the 7,000 languages that are spoken globally today will likely dip under 100. As we move even further into the future, the world's rising temperature and accompanying deterioration in the protective ozone layer will play a major role. Added UV radiation reaching the planet may create a scenario where darker skin becomes an evolutionary advantage as it protects against UV damage. Increasing temperatures may also affect our size. Taller and thinner bodies would be better at dissipating excess body heat as it creates the best body surface area to volume ratio. Of course, these types of changes take tens or hundreds of thousands of years and will depend on our ability to avoid or protect ourselves from nature. New traits from mutations may also arise, perhaps a new eye color or unique abilities. Even today, one man has been documented with the ability to consume and digest nearly anything, including metal, glass, and even toxic chemicals due to a genetic mutation. Others live with a condition called tetrachromacy, where they can see 100 times more colors than the rest of us. But these traits are only likely to persist if they have some selective advantage, so don't expect a mutant-like school for the gifted anytime soon. But artificial selection, or human-influenced changes, will likely drive most of our evolution. We'll modify our baby's genes before birth to eliminate diseases they may carry or include desirable traits for our offspring. But while it may make us smarter, stronger, and better looking, such genetic similarity or lack of human diversity leaves room for a single new disease of the future to wipe out the entire human race. In the words of Stephen Hawking, sooner or later, disasters such as an asteroid collision or a nuclear war could wipe us all out. But once we spread out into space and establish independent colonies, our future should be safe. Space colonization, it seems, is an inevitable part of our future. At the end of it all, we may even conquer death by scanning our brains, atom by atom, and transferring that information into computers. In this way, we could travel at the speed of light as information patterns, unrestricted by our bodies and requiring no food. Where our current human generational cycle takes around 20 years, a digital individual could replicate themselves in seconds or minutes. It's a brave new world out there, and yet
It is a brave new world out there. In fact, I just read today in the New York Times that they think that humanity's population size will peak around 2064 at 9.7 billion. We're around 6 to 7 billion today. And then that will dip down to 8.8 billion by the end of the century at 2100. I've always, the last few years, I've been telling students, you are the first generation that will get to choose when you die. Now, you may die in a car crash or something like that, but if you're just going into old age, you'll be able to recycle out all of your organs and anything that you need fixed years from now. So it is a brave new world for sure. Let's go on to theme 4. Continuity of life is based on heritable information in the form of DNA. So the idea here is that when organisms pass on information to other organisms, they help the incessant chain of life continue. And this is in the form of something called DNA. And you've seen this before. We have DNA in the form of these things called chromosomes. How many chromosomes do we have in each cell? You're right, it's 46. 23 from mom, 23 from dad. That's being shown there by the sperm and the egg cells. The idea then is that each chromosome is one long I keep looking at the screen because it's up, and it's what I'm used to. Sorry. It's one long DNA molecule with hundreds or thousands of genes. Remind yourself what DNA is, please. Remember, DNA is made up of nucleotides, such as A, T, C, and G. Oh, by the way, on the Google Classroom, I have taken a pre-picture of this board, which was empty. So that might help you. You can press pause at any time, obviously. I do want you to write. I believe in the value of writing. Don't just sit there, please. Obviously, DNA is inherited by offspring from their parents, and it controls the development and maintenance of organisms. What that means is that everything that we have comes from DNA. Now that could change over time or have little wrinkles on it, but generally speaking, the characteristics we have come from DNA, and it comes through this little process right here. You've seen this before. Watson and Crick called this the central dogma of biology, and all that means is that it's the flow of information within every cell, and then also by, I guess, extension from us to our children, because we give them these same bits of information that we call DNA. Here's what it looks like, cartoon form, a bunch of sugar and phosphate groups on the outside, with a bunch of these letters, A, T, C, and G in the middle. You've seen all those before. Remember, there's 2 single strands of DNA which come together to make that double helix. And there are special portions of the DNA called genes. Now, science believes we have about 21 to 23,000 of these. And these are specific sequences of DNA which actually do something. 99% of our DNA doesn't code for protein. There's tons of debate as to what it does, and people used to say it was junk, or it was viral DNA that was replicated, or it had no purpose. And what, of course, we're learning now is that it has tremendous purpose. That's probably the most dynamic field in biology is genetics, which is exciting. We'll get there this semester. But genes are important. There's specific sequences, chunks of information on these long stretches of 6 billions of letters of DNA per cell that actually do something. This is what they do. They are the messages which actually create proteins. Anyone remember what this process is? Maybe I should turn the arrow around here. This is, remember? This happens in the nucleus. It's transcription. And then this second green arrow, anyone remember? Oh, I just heard you say it, translation. So the point is that the information on the genes is transcribed into RNA, then that information is translated into a sequence of amino acids which fold and become proteins. Here's an example, here's an example. So there's some protein that we have in our eyes called crystalline, and this is a figure from your textbook. It says, how do lens cells make crystalline? Well, there's the gene. We have the DNA, we've got transcription. RNA is created, and then it's translated into those amino acids and it forms. So this is super important. I know you know this from 10th grade biology, but you need to rehash this. So go ahead and push pause and tell your favorite stuffed animal what you know about the continuity of life, how it's based on this inheritance pattern of DNA and transcription and translation. So another theme that we're going to go over that you're going to see in the first chapter is homeostasis. And basically, that's the retention of set points, or the desire for the body to go to certain set points, like your best resting heart rate, blood sugar concentration, temperature, salt concentration in the bloodstream, blood sugar level, all these things. These are feedback loops, and you see up here negative and positive feedback loops. You remember these from 10th grade biology. The point is that things are always sensing what's going on in the body and then feeding that information back and saying, all right, let's adjust and get it back to the right place. But life is filled with challenges, and you always see things happen in our environments. And so we're going to get out of whack, and then we're going to bring them back to the set points. And so this is how it does that. Through homeostasis. This is a picture that you see in your textbook. Go ahead and try to explain this to someone who's handy and there. Your friend who's on FaceTime with you right now. What do you see when you look at this? This is negative feedback. Let me finish this this definition here. Negative feedback is when there's an accumulation of end product, and that's going to slow the production of itself, or slow the production of the end product. You know, I heard this story about these young men in the trenches in World War I in 1917 and 18 in Europe, and they were in these trenches, and there were bombs going off. There were threats to them all the time. People were coming over with bayonets and trying to kill them, and the point is that they never were able to rest the whole time. What happened was these young men, 18, 19 years old, they got dismissed from the war. Maybe the war was over, and they would go back to live in their northern European country, like the Netherlands or Denmark or France or wherever they lived, and they would literally be a vegetable for the rest of their life. And what scientists now believe happened was that they were so stimulated all the time. They were doing a process and creating with adrenaline from the adrenal cortex of the kidney. They were creating all these things, these stress hormones and an ATP and all these things in their body, and they were never able to feed back and stop that process. So they were basically just zombies for the rest of their life. It's really sad. You know, you can imagine if you were laying on the couch at night and you hear a noise and you get scared, adrenaline comes into you and ATP is made, and there's a lot of ATP that's being produced, but then after a few seconds you realize, oh, okay, that's just the neighbor or the dog or whatever. Then you start to say, I don't need, so subconsciously you're saying, I'm producing all this ATP to do a fight, flight, or freeze mechanism, a sympathetic nervous system activity, and you're like, but I don't really need it anymore. And so what happens is as the ATP starts to build up, but it's not being used, it actually feeds back, and you can see that little negative sign there. And it actually, there's a slot inside of enzyme 1 where it feeds back in, changes the shape of enzyme 1, and then it stops this whole cascade from its own production. It's ingenious. I hope if there's one thing that you learn this year, is how wonderful you are and how wonderfully complex and awesome nature is. But the point is that it feeds back, stops its own production. Go ahead and try to think of an example. And I'll show you a little movie that came with the text.
This simple animation illustrates the principle of negative feedback, or feedback inhibition, in the regulation of a chemical reaction sequence in a cell. The sequence involves 4 molecules, A, B, C, and D. 3 different enzymes catalyze the conversion of one molecule to the next. The final product, D, inhibits the first enzyme in the sequence. When the concentration of D rises to a certain point, the reaction shuts itself down.
It's pretty cool. Here's an example from your textbook. This is a picture of how blood sugar rises after you eat. So you have a high sugar level, a blood glucose level. The insulin-producing beta cells in the pancreas will then secrete insulin into the bloodstream. That then goes all throughout our bodies, and it binds to certain receptors on the liver and muscles specifically, and then the glucose is taken up by the liver cells. But the point is, when that happens, it then feeds back and says, okay, stop creating more insulin because we've taken all the blood sugar out of the bloodstream. Well, here's the other example from your textbook, positive feedback. Go ahead and press pause and take a look at this for a moment. You can see here how the product is going to feed back in a positive manner. What does that mean? It means the accumulation of the end product actually increases its own production. When would this happen? Any ideas? There's two primary examples that are given in college freshman biology classes of this. Number one is in childbirth. When a woman begins labor, there's something called oxytocin, which is secreted from the anterior pituitary gland in the brain, goes to the bloodstream, goes all throughout the body, through the whole bloodstream, goes and touches every cell essentially, but it fits into a receptor on the uterine smooth muscle cell of the uterus where the baby is being contained. And the idea is that it binds to the receptor, causes a pathway to occur, and then causes a contraction. That contraction then releases a chemical which goes back up to the brain, which even secretes more oxytocin. A minute or two later, that thing gets down there, and there's even a greater or more often contraction. And the point is that every time a contraction happens, it feeds back, and then women, and you can ask anyone who's had a child, their contractions get stronger and deeper over time until the activity is done, which is to eject the baby from the uterus through the vaginal canal. Baby goes out. Then at that time, there is a hormone that's released that goes back and it stops the production of the oxytocin. So there's no more uterine protection. That's not true. The placenta has to pass also. Once the placenta passes, then that occurs. The other example is through blood clotting. The point is that once the blood clotting cascade occurs, if you get a cut and these platelets and these things start to agglutinate in that area, then that feeds back and creates even more of those same proteins until the clot occurs. That's why it's slow at first when you want to clot a cut, and then it'll eventually.
In positive feedback, a product of the reaction sequence enhances the action of one of the enzymes, increasing the rate of production of the product. Positive feedback is less common than negative feedback in living systems.
I'll go ahead and answer this out loud. How many species do you think that there are on planet Earth? Species of organisms. When I was a student, I was taught, this was a rough estimate, but people thought that there was 1 million species. By the way, as a side note, did you know that if you discover a species, you get to name it? There was a Glenwood High School student, this was a few years ago, in the Fairy Caves, which are these vapor caves by the Glenwood Hot Springs, and this individual was really interested in caving, and she found a spider way in the back there, and the spider had never been described by science before, so she got to name it. Well, before I answer that question, I want to show you this picture, and I don't know if this is the actual dude or not, but this is a process which occurs. So there was a Harvard professor, and this was about 25 years ago, and he took his graduate students, and he went down to the Amazon, and he went under these big trees in the Amazon, Brazilian Amazon, and he put these tarps around the bottom of these trees. He and his students for about 4 to 5 hours would smoke these trees. Essentially, they would have these huge smokers, and they would create tons of smoke. It would go up into the trees, and they would essentially try to kill everything in the tree that wasn't the tree. So they would turn off the smokers, they would walk away, and over 24 hours, with the little wind gusts and stuff, the insects and other organisms would rain out of these trees. So they would collect them on the tarps, and they would fold up the tarps, they'd pour everything, all these dead organisms, into like a Home Depot bucket. They would go back to their biological research station, and they would spend the next few weeks counting and sorting through what they had. What they found was for every Amazon tree that they did that to, they found about 1,000 new species never before described by science. Could you imagine trying to name all those species? The point is that he, at that time, him and his group, turned on its head taxonomy, which is this kind of nomenclature that we use to organize living things in the biosphere. And as a result of his work, they upwardly estimate now that it's about 15 million species that are on this planet, but only about 1.8 million have actually been identified. Well, the environment is facing a lot of trouble, and I guess you know what smoke does now, the Amazonian trees. There's also trouble with coral reefs, which are the other kind of biologically wonderful places on this planet. If you've never been to a coral reef, I recommend it. They're leaving us at a very quick rate, but they have tons of biology there. One of the reasons why is through there's higher levels of UV radiation now because the ozone layer is being broken down, like that movie said, and that's causing damage to the coral. But also, I have a chemistry equation up here. If you have increased carbon dioxide in a system that contains water, and de Chatelier's principles tell us that if you increase one side of an equation, then that's going to push the equation to the opposite side. So the point is that if we increase CO2 that's dissolved in water, it's going to push to the right. It's going to push to the right. We'll get more of this bicarbonate ion. But the point is, this is the problem. We're going to get more H plus ions in solution. And what does an accumulation of H plus ions equal? You're right, lower pH. And that's why when you hear the oceans are acidifying, and they're becoming more and more acidic, and that's hard on the coral reefs and a lot of organisms that are there. That's a problem. Well, we've had, science believes, 5 mass extinctions in the past. There's always going to be some death of species over time, but it's really slow. Right now, we have about a speciation loss of about 400 species per year on this planet. And it's usually like one species per like 8 or 9 years or something crazy. So different than that. And so much that science believes actually we are in a 6th mass extinction right now due to, yay, humans, and all the activities that we are engaged in. Well, you'll see this in your texts. You've seen this before. Domain, kingdom, phylum, class, order, family, genus, species. We'll get into this more in chapter 26. This is just like a rock skipping across water. We're just touching on some of these concepts, and we'll go into them in a lot more detail as this course goes along. Don't have to memorize all this right now. Just know that there's this organizing system. This is called taxonomy, branch of biology that names and classifies species. You can see here, your book's going to talk about how it's organized as far as bigger picture. Here's what I mean. When I was a student, I learned that there were 5 kingdoms, and in fact, that people used to think that there were 3 kingdoms. And the 5 kingdoms that I learned were plants, animals, fungus, protists, which are single-celled eukaryotic organisms, and then prokaryotes, which are bacteria. So what happened was about 10 years ago, scientists were up in weird environments, like in the salt flats of Utah. They were up in the boiling pools of Yellowstone, and they were finding these single-celled organisms like scientists have found in the past. You can just take a water sample, stick it under the microscope, and see things swimming around. But what they didn't do before 10, 15 years ago was they never sequenced the DNA. What they found actually was that the DNA was so similar, more similar to eukaryotic organisms like us in these single-celled organisms than the DNA was comparable to a prokaryote or a bacteria. And so what they found was this whole 5-kingdom system had to be scrapped, and now we have 3 domains. So domains is like even more inclusive. It's a larger body of organisms than even kingdoms are. 3-domain system is currently used. It replaces this old 5-kingdom system. Here are these archaeobacteria. Archaic means old or been around for a long time. Here's the prokaryotes, the bacteria that you know, E. coli bacteria in your stool, or bacteria in yogurt or cheese. Here are the 5 kingdoms that exist under the eukaryotic umbrella. These are all eukaryotic cells. We define those in the first part of chapter 1, main eukaryote. Well, The last thing we're going to talk about before we watch a movie on the scientific method is Chuck. I love Chuck. Chuck has the sickest sideburns of anyone I've ever. Those lamb chops. Anyway, you know Charles Darwin. He took a cruise around the world, and he did a lot of things, and we'll get to that in just a moment.
The geneticist Obzhansky said that nothing in biology makes sense except in the light of evolution. But was he right? Let's try to understand biology without evolution by natural selection. Why did Pavlov's dogs dribble? If an animal can learn to associate one stimulus with another, such as a bell and food, or a predator and danger, it will have an evolutionary advantage. Why does a hoverfly look like a wasp? The harmless hoverfly doesn't have to pay the cost of a sting, but gets the benefit of not being eaten. So, more wasp-like hoverflies have evolved. Why do antibiotics work? Penicillin and other antibiotics come from fungi, which have evolved resistance. Why don't my antibiotics work? Bacteria have encountered antibiotics so many times that any mutation that makes them resistant will be selected. Why does chocolate make me happy? In the wild, it's hard to get enough calories. Since sugar and fat are high in calories, we evolved. Why are trees green? Evolution. Why are birds feathered? Evolution. Why do we exist? Evolution. Get the picture?
All right, so there's two main points. I want you to write these down. He believed that with all of his journeys, and don't get me wrong, this is my favorite part of biology. We're going to study him chapters 22, 23, 24, and 25. So I'm not going to go into too much detail here. But he showed that species showed evidence of descent modification through time, like with this kind of evolutionary tree, where species of organisms are branching off from a common ancestor. The second point was that natural selection is the mechanism for this. Go ahead and write those two things. Turn to your favorite stuffed animal and tell them how the kangaroo's pouch came to be. How? Marsupial mammals. Here's a picture that I like to give to students and have them then explain it to their neighbor. And you can see there that there's beetles of different colors. Some are a little bit lighter in color and some are darker in color. See that populations have individuals that vary with traits. More individuals are produced and survive. There's always competition. But some are better fit in their environment than others. And then those ones who aren't are selected against. And then you can see the individuals that are selected for with their beneficial characteristics survive, and then they reproduce and have organisms with those beneficial characteristics. So over time, you can imagine that this will be reinforced, and we start to see things. We see a match between organisms and their environment as far as them being well fit in their environment. Here's some needle-like spines on the underside of an acacia tree. Go ahead and explain how you think the acacia tree evolved to have these needles. It's a pretty wonderful concept. Here's a picture from your textbook. I like this picture. It's basically saying the same thing. Picture that you memorize for the test. Evolution is the concept which follows the entire course. We always talk about evolution and why things are the way they are. Maybe we don't know the answer, but we try to appreciate that in every chapter. So make sure that you understand this concept. And on the exam, I'm going to give you examples like of acacia trees and marsupial mammal pouches and things like that. Then you're going to have to apply these steps to the example that I've given to you. I'm never going to ask you to regurgitate any information in this class. I'm always going to give you like a data or a table or a graph or an example, and then you have to apply what you know to that. It's hard. The tests are really hard. Some kids will tell you that. Well, here's finally 1.3. This is the scientific method. It's pretty simple. They give one primary example of a flashlight that doesn't work. Why doesn't it work? Well, there's one hypothesis. Maybe the batteries are dead. You can go through steps in order to see if you are correct with that hypothesis or not. So instead of me talking about this, let's watch a movie. This is my friend Alex from Tufts University.
Last week, Hurricane Sandy ripped through the Northeast, meaning that everyone went out and stocked up on bottled water, non-perishable food items, and flashlights. And the last one got me thinking about the scientific method. I remember back in elementary or middle school when I was first learning about science and the scientific method that my textbook illustrated the concept using the problem of a broken flashlight. This means that in college, when I was in lab and frustrated and angry about why my experiment wasn't working and my hypothesis wasn't right and what was happening, my data didn't look right, I would pause and step back and mentally go back to the problem of the broken flashlight to try and figure out where it got wrong. So I know that for some people the scientific method is a really basic concept, but it's really important and it's the foundation of every good experiment, and the broken flashlight is carried throughout my entire scientific career. People will break the scientific method down in slightly different ways, but I have broken it down into 5 simple steps. Step 1 is your question. Now every lab and every experiment begins with a question, and some of those questions are big and broad, like how does your brain work, and others are small and detailed, like how does myelin impact axon signal propagation in your neurons, but each of them is a question. Today, our question is why won't my flashlight work? Step 2 is background research. So before you go barreling into an experiment, you want to find out what other people have done before. This way you can make an informed guess about what might be the problem and what ways you might try and go about solving it. Step 3 is your hypothesis. So after you do your research, your next step is to make an educated guess about what your problem might be. I found in my research that sometimes if your light bulb is broken, then your flashlight won't turn on, and that makes sense to me. It is very possible that I have a bad light bulb here. So that is my hypothesis. I think that my flashlight is not turning on because the bulb is bad. Step 4 is experiment. It's probably the coolest step. So now that you have your hypothesis, you have to go about testing to see whether or not it's right. So I'm going to replace the bulb in my flashlight and then turn it on and see if it works. Now it's important that at this step, all I'm doing is collecting data. I'm not making any judgments. I'm not coming to any conclusions. I'm just seeing what happens. And the flashlight still doesn't work. Step 5 is analyze. And it sounded cooler. It's just analyze. So now we look at our data, try and make sense of it, and move forwards. So I saw that my flashlight did not turn on after I replaced the bulb, and therefore I'm going to conclude that the bulb was not my issue, or that if it was not my only issue. So now we're done. No, we are not done. If science just stopped every time a hypothesis was wrong, we would never get anywhere. No, now we go back to step 3, hypothesis. My first hypothesis was wrong, so I'm going to make a new one. I think my flashlight might need new batteries. Well, what do you know? Go for it. Do science.